What I'm going to talk about now is so strange that I wasn't going to talk about it. I was going to ignore it, even though this was a peer review published paper. But then something equally strange happened with regards to Health Canada that really got me to thinking. Now, for anyone who has listened to my podcasts and videos, you will know that I am no stranger to conspiracies or controversies. In fact, I actually look carefully to see whether or not science could be accurate. And I've been following that line for quite a long time. And what is very worrying is that I can't think of any one of those things that weren't actually true, which is why I had to ask my question again. Could this question about vaccine nanobots actually be true? It's really strange. But I'll share with you the paper and allow you to come up with your own conclusions. But importantly, as I said, because I am not afraid of asking these hard questions, you will know that I've been following the other strange story about Embalmers clots. I've recently been given very important information about the composition of these clots. And I plan to talk about it in a webinar in another few days. So please register for this because this, in my view, is extremely important. Because when I looked at the composition, I had a brilliant idea. I think I know what this is, and I think I know how it occurred before. Now, it's just going to be my extrapolation of the science, but as I said, there is always something that needs to be analyzed in order for us to make sense of what's in front of us. So, again, as I said, this paper was just a little bit strange, even for me, and so I stepped back from it. So, here is what the paper is about, and I'm going to give some context to what is happening here because if you mention this in the mainstream circles, I know exactly what they will say. They're going to debunk it immediately. Don't care that it's peer reviewed, the journal is irrelevant. So, this is what you will see. This is the International Journal of Vaccine Theory, Practice and Research. And this paper was published in July of 2024. And they were looking at self-assembly of stereomicroscopically visible artificial constructs in mRNA products, mainly from Pfizer and Moderna. They did a longitudinal study. Okay, as soon as I saw the paper, I thought to myself, there is no way that would be published in a mainstream journal. Just won't happen. They would just refuse to do it. And so you'd never hear about it. So this then got me to thinking about what is happening in terms of science. When you have voices that are outside of the standard thinking, they get silenced rather than discussed and debated. And what then happens is that those bits of information then get put in alternative sources of media because it still needs to get out. And so when you look at the journal that it was published in, what I did quickly is I just went and looked at their history, and I can see from their archives that they had been publishing since about 2020. And they started off with global vaccine controversies. So straight away, they recognized that there was no easy way to get out challenging information in mainstream publication. So they made their own journal for which they'd have their own peer reviewers. And they have been publishing since then. These are all their journals. And coming up here, this is in 2024, where they published this actual paper. So what you will find happening is that if you mention the article or the paper, those people who are against or with that narrative will say they will ignore it completely because it is not from a reputable journal. I understand that. and we have issues with these things in the scientific community, but it doesn't mean that the study has no relevance. That's where I am different in that I would say, okay, I accept that this is not Nature or the Lancet, but it doesn't mean that you discount completely what the study is looking at, if that makes sense. So, again, what you start off with is you look at the authors of the paper. And you would start off here and recognize that when we look at Dr. Young Mi Lee, this person, obstetrics and gynecology, and this is in Korea, South Korea. And you have Daniel Brody, which is a professor of applied linguistics. This is in Japan. Now, the purpose of the professor is more to do with setting up the study and making sure that it is following appropriate approved standards in terms of the way the research is being done. So the fact that somebody is not necessarily scientific could actually give credence to the fact that they are actually trying to look at this objectively. So I'm just telling you straight up front why a lot of people in mainstream will just completely ignore this information. However, what I realized when I looked in detail at the paper is that they did a lot of research for an extended period of time. And that's not easy to replicate if you are looking at something over a long period of time. We're talking about certainly over a year, maybe 18 months to 2 years that they were studying this, looking at the vials and so on and trying to make sense of it. So it's not easy research that they will have to replicate. And what it is that they're doing is what I would consider should be done by regulators. You have to remember that people who are trying to find problems, they're not bad people. They are good people. They are people who are making sure that what you're being given is actually safe. So they're digging around trying to find problems to see whether or not this is actually true. If somebody comes out with a new drug and they say this is wonderful, you could pick any of the new drugs that are coming out being promoted, and then somebody goes and starts to search for all the issues that could be relevant. That's not a bad thing. That's a good thing. That's good prudence, because if they can't find anything, that is a good sign that the product is safe. And so, therefore, everybody benefits. The person may have wasted their time, but there are no issues that are concerning. And so, in that sense, it highlights that this kind of research is still very, very important. And as just mentioning before I go on with this, remember that our Kickstarter campaign is on. We are at 67% of our target. Are you prepared? About Disease X, which in my view is really an extrapolation of COVID. And this is our book that we're putting together. Critically, what we're doing as well is that we have added a section in it. This is not a section in the book, but just as a sweetener, an excerpt from Capital COVID Connections with Dr. Malone. And this here is just a summary of the things that he had said at this this Capital event, where he put forward the science and just laid out what his concerns were as somebody who first thought about the mRNA technology. I'll come back to it again in time, but it's useful for you to have at your fingertips just a summary of the information that he was presenting. So let's get back to this paper. I'm going to highlight a few things that were said in the abstract of the paper, just so that you understand what they were going for. So let's take a look again at the paper in a little bit more detail. So I've highlighted a few points here. And the first thing is that they got carefully preserved specimens, were cultured in a range of distinct media to observe immediate and long-term cause and effect relationships between the injectables, the living cells, and living cells under carefully controlled conditions. So, this is why I said it's not easy to replicate because they did this over a long period of time. And they were looking at vials that they got from Pfizer and Moderna, as well as AstraZeneca and Novax. And they also highlighted here that what they found were animated worm like entities, discs, chains, spirals, tubes, right angle structures that couldn't easily be explained in these vials. And they've come to the conclusion that it suggests there is some kind of nanotechnology in the injectables. That's their conclusion. It's not a fact. But they are saying that they are noticing some patterns in it that are abnormal that they haven't seen before. And so, again, it is not something that you want to completely ignore. It does warrant at least a bit more detailed analysis. What is it that they could be seeing? Because it's one thing to say they disagree with the research, but you still have to explain what it is that they actually were seeing throughout these products. And so I'm trying to find in the paper what they did is that they collected samples. They got two whole vials. I'll show you it here. It shows you the materials and methods just to show you what they did. So they have here 54 samples. They had 50 residual injectable vials. So that means that they got vials, 43 of them Pfizer, 7 Moderna, acquired immediately after their use in the COVID vaccination campaign. So this was the residual vials where it had little bits of residue left in them. And they also got 4 unopened new injectable vials, 2 Pfizer, 1 AstraZeneca, 1 Novax, which they studied. So I think one of the questions straight away is that if these were at the end of the 6, say for instance, each vial can usually give about 6 vaccines. Were they picking up residue from the manufacturing process? Is it more likely that the residue would fall to the bottom? But in truth, when you think about how injections are done, you tend to turn the vial upside down so that you can pull out the fluid and then inject it. So you're continuously moving the fluid around. So it is less likely that you're going to have residue at the bottom that is left that wasn't there before. So it is certainly possible. And I'm sure that's one of the things that critics would debunk about what it was that they were doing. This here is the image of their facility, and they were using microscopes and collecting data and so on in a clean environment to get this information. I can tell you from firsthand experience when I speak about the embalmers clots, the scientists who are doing this are oftentimes doing this at great concern about their professional um, um, abilities because they will be targeted because of it. And so when we did well, not we, when I was told about the embalmers clots, the scientists who were involved made sure to say that this information needs to be kept under wraps until the work is done, because they were worried that if people knew who they were and what they were doing, they would stop the research. I suspect a similar thing could have happened here in that this was done very quietly. They went about getting all of their information, put it together, and then they published it. So it is concerning that this is how scientists have to operate. And, as I said, people don't think censorship is real or this kind of information would be suppressed. No, absolutely. It is being suppressed. It is having an impact with regards to what people are seeing and the kind of research that can be done. I don't know how we change this because it has real implications, not just in the context of COVID, but it really has to make us ask questions about what is it that we are agreeing to as good, strong, peer reviewed evidence? Is it actually been contaminated through finance? Really big questions that we have to analyze in relationship to that. It's a very big paper, and I'll show you here a few more images as to some of the things that they were seeing in the microscope. And so these are real things that they are seeing when they had incubated them. I mean, this kind of pattern sounds like contaminants that could be part of, maybe this was in the manufacturing process, pieces of metal, I don't know. But it's not normal. And these are the kinds of things they were seeing in the vials. Okay, and I'll come to what the relevance of that may be just in a short time. But let me tell you something else that was very, very strange. That, as I said, caused me to really think carefully about the relevance of what this is. And it's you probably haven't heard about this, but it's a very, very important story that I'm making sense of. I'm going to make this a little bit bigger so you can see it. Um, so I'll just add this in so that you can take a look at this. This is an article that was published just recently in Ottawa Citizen. And this is the 4th of September that this was published. And Health Canada orders existing COVID 19 vaccines destroyed before approval of new vaccines. It's really strange. And so they're making the point that this leaves Canadians with no available COVID vaccines until October when the new ones come out. And, this is in the middle of a summer wave. They're trying to understand. So, Ontario's Chief Medical Officer for Health, hold on, let me make it a bit bigger. I think that's a little bit better for you to see. So Ontario's Chief Medical Officer for Health, that's Dr. Kieran Moore, sent out a notice saying that they had issued a market withdrawal of all COVID-19 XBB vaccines in Canada to take place on the 1st of September. And it was because of a contractual agreement between Health Canada and vaccine manufacturers, according to government sources. That was the reason that they gave at the time. And that new and updated vaccines for Pfizer and Moderna cannot be distributed until the remaining supply of older vaccines is withdrawn and destroyed. Had to pause there a second as I tried to get my other screen, a little bit smaller. So yes, you can see what happened there, and it's really strange. Why would they destroy them? You know, why would they stop giving them by the 1st of September? What is it that they know that we don't know? And why is it that when you consider the other side in the US, the CDC is now promoting these XBB vaccines that children under a year must have three by the time they're a year old, so by 9 months. And so the whole picture just doesn't add up. And I went all the way back to thinking about the fact that Health Canada had been given specific information about contaminants in the vaccine very early on. And I think Professor Byron Bridle had done this. He actually lost his job over, I think, raising some of these issues. But they had specifically been given this information. So it makes you wonder whether or not it's possible that what is going on here is that they recognize that there may be issues with contamination. They want to wipe the slates clean, and the regulators want to bring in new vaccines that don't have any kind of contamination in it, because now information is coming out about what else could be in these things that could cause problems. So that's one side of it. The other side of it is this along the lines of what happened with AstraZeneca vaccine, where it just stopped being used and then eventually it was withdrawn. And if that's the case, what you will find happening is that the vaccine will disappear for some months, and then in October and November there will be issues with supply and you just can't get enough, or and then it just won't be used. And it would therefore indicate that maybe something is happening in the background, and therefore you do have to wonder whether or not this is all relevant. Everything is tying together. So it may be a bit of a stretch from a conspiracy point of view, but it is an important point, I think, that made me reflect on it. Here are some of the other things that the paper found. And this is relevant when they were looking at the injectables. So in this picture here, they have Pfizer, Moderna, Novax, and AstraZeneca, all four vaccines, and they are looking at the plasma reactions. So they added it to normal, as somebody who is unvaccinated, their plasma. And they found in the Pfizer one, there was cellular collapse of white blood cells and damaged platelets. This one here, Moderna, roller formation. This is where we use things like an ESR, makes it higher. This one here in C with Novax, the nucleus of the white blood cells were disintegrating. And in this one here with AstraZeneca, again, you can see the red blood cells here are clumping together. And so it would make the elevated ESR. So there are changes that occur in the plasma at the time that could or probably would not be considered necessarily normal patterns within a short period of time. They tested it with sperm as well. As I said, they did a lot of tests over the period of time. And what they did is they used a number of controls, and I'll show you here again. Um, they tested, this is Pfizer, Moderna, AstraZeneca, Novax, a flu vaccine control, and normal saline control. So they tested all of them 10 minutes, 30 minutes, 1 hour, 2 to 3 hours. And this was semen from a 47-year-old. Within 3 hours, it was 0% motility, 0.1% motility with Moderna. Compare it to normal saline control, remains stable over that period of time. But saline would represent a normal media for sperm to be in. Even the flu vaccine control, there was some reduction, but not as significant as all of the vaccines. It was 0% within about 3 hours for all of them, stable in normal saline. Now, again, somebody debunking it would say, well, in any case, what would you be putting the vaccine with motile sperm about? It's not relevant. I think the point being, though, is that if you had circulation of these products, you could then have damage to cells, which is one of the questions that hadn't been properly answered in terms of biodistribution. The other thing that they looked at here, they were looking at these geometric entities that they saw in the paper. And what they found, and this is why I said it's not easy to replicate. So they were looking at this from week 1 all the way to 10 months. And they were observing this under the microscope. And you could see that these things peaked by about 4 months. It increased up to here and then gradually went down. And each one of the colors represents, so one is dark blue is Pfizer in normal saline, orange is Moderna in normal saline, Pfizer in distilled water, Moderna in distilled water. So they just used different controls to try and see if there was any pattern. But essentially, all of them, it increased for about 3 or 4 months and then gradually decreased again. Really, really strange patterns. This is what I'm saying is that whatever anyone may think about the journal that it was in, there are just some things here that they have done that are not easy to replicate, that you need to go and look at in more detail to try and say, is there any other explanation for some of these patterns? And again, you have here, this is another image showing again time frames. And you can see here again, this is over 12 months they were monitoring this in distilled water, normal saline. And you can see the red one is Moderna in calcium hypochlorite. No, that's the brown one. The red one is Pfizer in distilled water. And you can see here that this just gradually increases the approximate numbers of developmental grade of entities within the microscope. So it gradually increased over a 12-month period of time. Makes no sense. It really, really is strange. And even if they had contaminants in their system, you can see that when they use the control, some of these patterns did not occur as well. So again, I'm going to show you some of the strange things they noticed under the microscope. This is the kind of shapes of things, these spirals. Really, again, you would think that these are probably shards of metal. What is this? What are they? We're not sure. I genuinely can't make sense of what it is, but I don't discount what they saw because they took the pictures of it and they were doing it for such a long period of time. So it really is unusual patterns. And again, here are some more images with the paper that they took. And these are on the very powerful microscope, so you wouldn't see it otherwise. But what are these things? Very, very well, they're not randomly shaped. And so this is part of the reason why they think that this is relevant. This is not just a contaminant because of the unusual patterns and shapes that they take. It's hard to know if this was ever done before in any other product, and so we don't know if this is necessarily happening. But it's important to note they only saw this with the mRNAs. They didn't see it with the AstraZeneca and the Novax. It caused other problems, but it didn't seem to be there. I'm going to show you another thing that they did. And this is the thing that, again, this is really too strange. I mean, the what it means, I genuinely don't know, but it's now aligning with some of the weirdest conspiracies that I've heard that genuinely I just ignored. But here is one of the things that they did. And so clearly they were listening to the conspiracies as well, and they decided to test this kind of stuff. So what they then did is that they then tested it with EMF, wireless recharger with a cellular phone. So they put a culture dish with it, set it to 5G streaming mode over a wireless charger for 1 to 2 hours. And they were taking electric and magnetic measures through the triphase field, measured at 300 volts, and they were measuring it. And so they did this for 1 to 2 hours. This is what it looked like at the start. This is what it looked like within 2 hours. Now, this is in distilled water. I have no idea why this would change so dramatically. They said the floating materials abruptly became larger and more numerous with sharper and more rectangular edges. And they said, in contrast, the Pfizer showed no immediate response, but instead, after 1 month of exposure, exhibited a delayed effect, a moderate proliferation of floating filaments, as seen right in figure 24. This is, I don't know what to say. But one thing is for certain is that this pandemic has brought out some of the most unusual things that I have ever seen. And I think certainly from my research in autoimmunity, I genuinely can't understand why the scientific community is not seeing what I'm seeing in terms of autoimmune responses. Why are they not concerned? One of the points that people say to me is that my research has largely been ignored because the implications are so serious and people understand it. That's why they ignore it. And in the same way, when I see this kind of strange pattern in science that is occurring that I can't explain, I still want somebody to look at this and corroborate whether or not these changes could actually be true. Now, if vaccine vials are being destroyed, it takes away the opportunity to do those kinds of detailed studies. And I would think and hope that with this kind of research being published, somebody somewhere is holding on to some of those and saying, let's do other checks and see whether or not this can be accurate. If we don't do this, and I said, I take us all the way back to the original question when we had a virus that looked as though it came out of a lab and the whole world largely ignored it. It's the equivalent of seeing someone doing something wrong. You ask them about it, and they say that what you saw was not what you saw. And then you believe them. It's almost guaranteed it will happen again. And so the lesson that I have seen coming out of the pandemic is that whenever there are any unusual or strange patterns, we have a responsibility to ensure that there is nothing wrong that is happening. And the more people that ask these questions, the harder it becomes if somebody has any alternative agendas. And you have to remember that this kind of issue, when you think about it, will very often not actually benefit the people who are executing on what they are doing. They're just mere pawns in this. And we have to somehow get further answers, get better clarification, and not be afraid of conspiracies. The point of a conspiracy is to make sure that any element of it can't be true. As they say sometimes, where there is smoke, there is fire. But the fire may not be where you thought it was, but you still need to check where the smoke is coming from. That's how I look at it. You look at what is being seen, then you try and make sense of what it is that is in front of you. That's what I am doing with regards to Embalmers Clots. I think I have a good idea as to what could be happening, why it's not necessarily being seen by everyone. So if you want to hear what I have to say, join me in this webinar. It's coming up just in a few days, and I'll share with you my thoughts as to what it is that could be going on with that. Remember, that story is also considered a conspiracy. For me, when I now look at the details and the analysis, is not a conspiracy. This is actually an important piece of information that really needs to be assessed. So, as a final thought, remember, join us on our Kickstarter. We're very appreciative of any support that you can give. Help us to reach our target. And in the process, you'll also get a gift about Malone to learn more about what he said at the Senate hearing. And the final point for those people, if you are on YouTube, we've opened up a membership point where it's just easier for me to be able to answer questions. And so what I'll do is sometimes people say some very interesting things. It will be in that membership video section. A way of supporting the research is to join at a really nominal fee and support us as we continue to bring you cutting edge information, real-time science and analysis in ways that you've never seen it before. So really appreciate you staying with me here. This is interesting stuff. Share it. Get people to think about it. See if they have any ideas as to why this is happening. We need a few more conspiracy theorists, it seems, because everything they come up with these days seems to be coming true. Have a great evening.